Hi, this is Dr. Jabali, and this video is talking about topic 1.5. We're going to look at some questions from previous IB exams, written answers, basically long and short answers. Now, this first question here, I already did this in one of the previous videos, but I'm going to do it again because it's also part of 1.5. So this question is asking you to describe how Pasteur experiment provided convincing evidence to falsify the concept of spontaneous generation. So when you see this question, one of the most important thing is talk about Pasteur experiment. But first, what is the concept of spontaneous generation? So spontaneous generation is just basically means that this cell here, this new cell that came out of nowhere, it basically before it there was nothing. It was completely empty. So it was by magic. That's why it says spontaneous. It just appeared out of nowhere. That's what a lot of scientists believed back then. But then came Pasteur, and Pasteur, through his experiment, which is this one right here, he proved that that's not true. What Pasteur tried to explain is that any new cell that comes out, arise out of, it's actually coming from a previous cell that was living. So this living cell right here made this new cell. So for him, Pasteur, he's basically saying there's no way that this new cell could have come of nothing. This new cell came actually from a pre-existing cell that eventually divided and made and formed the new cell. Now, he didn't just say this. He actually went through the experiment to prove that. To go to the experiment, he basically had to use flask, and he had to use broth, and he had to boil it and manipulate the microbes that were going through it. So basically, when you're describing, one of the things you have to mention is you have to mention about the flask. You have to mention about the broth inside of the flask. You have to mention about the microbes or bacteria that grew on top of the broth. You also have to mention about the fire that they used in order to sterilize the broth. That means kill anything that is living there and make them all dead, right? Dead cells. It's like sterilize the same thing as clearing up, make sure it's completely clear away from any bacteria or any kind of microbes infection. So this kind of language, flask, broth, microbes, heat, sterilizing or boiling, these words need to be in your descriptions. Just quickly, I'm going to go through the experiment as a refresher for you. So basically, you have an idea of that. But you can also go back to the lecture video. So what he did was he put a flask, and in that flask, he put a chicken broth, and then he let the flask just loose. And out of the blue, after 24 hours, he starts seeing growth of bacteria on top of that soup. So for many scientists, this is known as spontaneous generation. See, those microbes just came out of the blue. Well, what he did was, the second time, he put the chicken broth again. Yeah, but he says, you know what? You see these little bacteria here? I am actually going to boil it. And when I boil it, I'm going to sterilize. And the word sterilize here is trying to say is that I'm going to clean it up. When he cleaned it up, there was no more of those microbes. There was no more bacteria. It was just the broth. So that means everything that was here, it was dead now. Dead cells. Now, he kept this thing for a day. Unfortunately, the next day, new microbes showed up. So he's like, this only continues saying that things come out of nowhere. But he really believed that these guys, right, they came from the outside. They came from the air. That means the air was contained with a bunch of microbes, and these microbes just landed here, and then they start growing their little growths of bacteria. So what did he decide to do? Well, he's like, I need to prove that these people are wrong. I need to prove that the spontaneous generation is wrong. So what did he do? He decided, you know what? I'm going to put a flask, but this time I'm going to close the flask, right? First, of course, he put the broth here. He closed the flask, and then he basically sterilized it. And then guess what? Days later, the flask was still closed, and guess what? There was no bacteria microbes growing. So right there, it was like, see, guys, there's only dead cells here. There's nothing inside. And guess what? None of the bacteria grew. So see, You can't just have things pop out of nowhere. They have to be popping because there was a previous living cell in there. So they say, how are you going to prove this? Well, let me show you this. If I crack it open, oops, that was too much. Let me go back. If I crack it open, I believe that the reason why you get these bacteria inside like this is because they came from outside, and outside there was those living bacteria that just came and landed here and made new cells. So it's not like a new cell just show up out of nowhere. These cells that showed up here in the bottom, they actually came from the living cells that were in the air. So he just disproved spontaneous generation and proved that the sentence from cell theory says the cells come from pre-existing cells. So that's what the experiment's about, and that's what the question is asked. Now, let's look at the mark scheme for it. So first thing says spontaneous generation is a life appearing of nothing. So first explain what spontaneous generation is and how this has nothing to do with cells coming from pre-existing cells. Then, of course, talk about the chicken broth, right, that was used. And so you say that chicken broth was put in a flask. You describe in the experiment. Then mention how the broth was sterilized or boiled to kill the microbes. Then say how after that nothing showed up. There was no sign of bacteria growing, right? There was nothing because it was sterilized. And IB allow you to use the word bacteria instead of microbes, okay? Then after you break, then after the neck were snapped, boiled, that means after you break the thing and allow, the bacteria from the outside to come in, what happens? Growth microbes happens. That means once you open that flask and allow the outside microbes to come in, growth started happening again. And when this happens, as I said, the cell, basically the broth became contaminated again. This just prove again that spontaneous generation doesn't exist. Now, this last one, it says curved necks allowed indirect exposure to air but prevent the entry. This, a lot of students don't understand what is this, but it's because teachers usually draw this. Oops, let me do it again. This is too much. Usually what they do is they draw a flask that usually like this. But when we say curved necks, we're talking about a flask that looks like this, and then it has a curve like that. So when you have a curve like this, the microbes can try to come in, but they will land here. They cannot fly up again and they go down to get into the broth. So these curved ones, they usually land in the bottom and they never reach the broth. Okay, while here they can just go straight in. That's what it means by the curved necks. Now that's way answer for the Pasteur experiment. Talk about the flask, talk about the broth, talk about the microbes, talk about the boiling, talk about breaking, opening the flask and allowing new microbes to come in. Mention this description and mention what spontaneous generation is and how it was debunked, and you get those full points. Now here on this slide, what I did was put all the questions that have to do with endosymbiotic theory. And believe it or not, every single one of these questions is asking for the same thing. But many students think they're asking for different ways or they're asking something else, but believe me, they're all asking the same thing, even though sometimes their mark schemes might be a little bit different. But what I did is that I put all these questions together and I joined the mark scheme for all of these guys. So whatever they had different, I put it in, and whatever they had the same, I did not add it up as two of them. So let's look at these questions. They're all the same. It says, describe the origin of eukaryotic cells according to the embryonic theory. This one says, apart from the ribosome, explain the evidence for the embryonic theory of origin. Again, this one was just saying, describe the origin. This one explain the evidence, which is exactly the same thing. Describe the endosymbiotic theory. This is very straightforward. Discuss the evidence of the theory of mitochondria that may have evolved from free-living prokaryotics by endosymbiosis. This is more detail. So you can see that the first one was very much more generalized. This one gave you a little bit more information. It gave you mitochondria, and it told you that this was evolved with free-living prokaryotics. And then the one says describe briefly. The difference between briefly endosymbiosis and this one is most likely that this one is asking for maybe 8 points and this is maybe asking for 4 points. But all of these questions are asking about the same topic. So if you have a generic mark scheme for this, you nail the points in all of these. So let's look at it. But before we do, let me explain for those who forgot what endosymbiosis means. The endosymbiosis is basically saying that mitochondria, right here, and with its second layer right here, as well as chloroplast, right? Chloroplast is right here with its second layer, right? And then he has his thylakoids right here. Now, both of them also have ribosomes, and both of them also have their DNA, right? I'm just going to put it as blue here. These guys are assumed that the two of them, they are exactly like what? A bacteria. Bacteria has a double layer, and it also has the plasma membrane, and it also has the ribosomes, and it also has the DNA. So they're assuming that mitochondria, as well as chloroplast, these two used to be bacteria. And what happened is basically a cell engulfed these two. That means the cell kind of like ate it, but I do not say ate it because ate it means you're digesting. So you just engulf them means you brought them in. And when the mitochondria got inside the cell as well as the chloroplast, they both became part, let me put here, or is it green? They both became part of the eukaryotic cell. And the question is, why would you have both the mitochondria and the chloroplast gets inside the cell? Because maybe the earth environment was so harsh, and for these two to survive, they said, hey, let's join our forces with this cell, because the cell can protect us. At the same time, we can give them energy and we can give them light, photosynthesis, all this stuff that they need. So it was like a symbiosis relationship. Hey, you give me this, I give you that, we benefit for each other. Now, where did they come with this idea? Why would they say that, well, bacteria, mitochondria, chloroplast are the same? Because there's a lot of things in common between these three, right? If you really think about what they have in common between a chloroplast, mitochondria, and the bacteria, there's 5 things, and you need to know those 5 things. 1, all of the 3 of them have their own DNA. 2, all of them have ribosome, and here's the best part about it. The size of the ribosome is 70S. Even though mitochondria and chloroplast are an animal, which have an 80S ribosome, inside of them, it's 70. Third, they all have a double layer. As you can see right here, right? Double layer right here. There's a double layer. There's a double layer. Fourth, they are all susceptible to be killed by antibiotics, right? Because we know the antibiotics are used to kill bacteria. But guess what? They can also kill mitochondria and chloroplast. And of course, last but not least, all of them, when they do cell division, they do with binary fission, which binary fission is the cell division of bacteria. But guess what? Mitochondria and chloroplast can make a copy of cells through binary fission. So these are the 5 evidence that might say, hey, it is possible that mitochondria and chloroplast was eaten by another eukaryotic cells, and then it became the big eukaryotic cells. So now that you have a refresh of what endosymbiosis theory is, let's look at its mark scheme. So the first thing the mark scheme says here, mitochondria and chloroplasts are similar to prokaryotic. That's what I've been mentioning the whole time. Second, theory that eukaryotic cells evolved from prokaryotics. Makes sense. We always think that prokaryotics came before the eukaryotics. Or it says, or cell took another cell by endocytosis, or what I was using the word engulfing, right? So that means the cell engulfed the mitochondria and the cell engulfed the chloroplast. Now, you're allowed to say take in instead of engulfing, but do not say digest, because digest means you're breaking it down. You're not breaking down the mitochondria, you're not breaking down the chloroplast, so do not say digest, okay? That will not give you the points. Then next, what we have, but did not digest. What I was telling you, you want to keep mitochondria inside alive. So this comes a symbiotic or mutualistic relationship. When I say mutualistic, that means we both benefit each other, okay? And then chloroplast organisms were once independent free-living organisms. Yes, we're saying that they were living outside of the cell because they had their own things, right? They had their own DNA, their own ribosomes. They didn't need to be in there. They only got inside of the cell because the environment was having hard conditions, according to this theory. DNA, they both have DNA. They both do division, binary fission. They both have the double membrane, right? Which is just like with bacteria. They also have the 70S ribosome, just like the bacteria. They both reproduce separate from the host or cell replication. Again, see, the mitochondria and the chloroplast, they can do replication. That means they can make a copy of themselves, and they don't need to wait for the nucleus to help them because they have their own DNA. Have organelles similar free living to free living prokaryotics. So basically, this is saying that prokaryotics, you never see a prokaryotic having an organelle. Why? Because the size of the mitochondria as well as the chloroplast, it's exactly the same size as the bacteria. So they're just free living, like, so basically we're saying the mitochondria and chloroplast, they used to be these prokaryotics that just live out there. They were just bacteria that live free out there. That's what we're trying to say. Again, have organelles similar to free living prokaryotics. We're just basically saying that some organelles can go out there living on their own. And then similar size to free living prokaryotics, what I just mentioned right now. And then you can see here the mitochondria in the membrane manufacture ATP like bacterial membrane. That's interesting. Some of the bacterial membranes can produce their own ATP. And guess what? The membrane of a bacteria is very similar to the membrane of a mitochondria. So they're both producing ATP, which again, that shows a lot that it's similar. And today, currently there is no free living prokaryotes like mitochondria. Today, you don't see any more mitochondria living on its own. You only see mitochondria inside the cells. What does that mean? It means sometimes in the old days, the earth had some kind of atmosphere that allowed the mitochondria to live by itself. But now that the earth atmosphere has changed, most likely now today's mitochondria cannot live on its own. It would definitely need another cell, an eukaryotic cell, surrounding it. Unless eukaryotics is evolved from prokaryotics. This is the most simple thing that Susan put, but yeah, eukaryotics evolved from prokaryotics. So this Mark scheme here, it sums up the entire process of the endosymbiosis theory. So it is very important that if you mention just here, if you look at here, if you just mention the 5 evidence, binary fission, double membrane, ribosome, let's see, where is the other ones? If I can have a double A ribosomes. It did not say, but here, it doesn't show where. I'm trying to find it here. Maybe I forgot. Okay, and say they're similar, but it's not there. I'm gonna find it. Oh, here, DNA, which right in front of me. The DNA, okay, that's already 4 over there. It doesn't talk about the antibiotic, but you can say, for instance, that they're just similar in living like a free living prokaryotic. So if you mention that they are killed by antibiotics just like a prokaryotics, that will consider as a point. And of course, they have the same size, so that's already 6-7 points. And then you can also talk about how they have multiple relationship. That's 7 points. And the points can just keep going on when you say with this one. So it's like you can get easily, just mention, first you mention the 5 evidence, then mention how they're similar to prokaryotics. So you mention how they went from prokaryotics to eukaryotics. That's like 7-8 points, and you nailed this question easily. So here's the last question. This question has not been asked by IB. It's not a past IB paper, but I decided to put because this is in the content, and I'm like, I have a hunch that this question will start to come up. So why not talk about it? If you remember the description of the experiment Miller and Urey, this guy, what he was doing is that they prove that, well, back then they used to think that organic molecule, right, can only be produced by a living force. So it's like the vitalism. However, these guys, they prove that organic material, okay, or molecule, can be produced by inorganic, which is not living, organic material. One specific example for this is urea, which is what you pee. Urea is what you found on your piss, right? So they used to think, well, you cannot produce urea outside a living body. They thought that urea can only be produced by something that is living. However, in the experiments, they actually proved the opposite. They did prove that you can actually produce organic molecules from things that are not living, in this case, inorganic materials. So they said, my friend, you can't produce a piss in the laboratory without needing a living being. That was the entire experiment. So with that, let's look at what are the things they might ask you on the mark scheme. So it's a non-living synthesis of simple organic materials has been demonstrated by that. So basically they're saying the non-living synthesis, that means you produce urea, which is an organic material. Now what they did is they recreated conditions of the earth back in the old days with using what? Flask and tubes. Now what happens? The water was boiled to vapor. That's why, because there was high temperature back then, right? Earth was very hot, so they boiled the water to make a lot of water vapor. Then what happened to that vapor? They mixed that vapor with different gases such as hydrogen, methane, ammonia, all this stuff, because that was what was the atmosphere back then. So you want to make sure that you're creating that atmosphere where there was no oxygen. So water vapor was mixed with these elements, these compounds. And then back then, remember, there was no oxygen in the atmosphere. When this mixture which is a mixture of water vapor with these guys, passed through electric discharge. Why? Because there was a lot of what? Lightning. There was a lot of lightning back in the earth. So the lightning is hitting in this mixture. Guess what that mixture came out to be? After that, the mixture, after it's been hit by the lightning and cooled off and left there for maybe a week, look what it came out to be. At that time, it just turned out to be traces of simple organic molecules. In this case, it was actually the piss. So here it's written, after time, condensed mixture was analyzed and found to contain traces. What it's basically saying is, taking all this sentence here, it says that it produced urea, which is an organic molecule. So urea was produced by using water vapor and these elements together to make urea. And that's what the Miller-Urey experiment is. So that's pretty much for 1.5. It's a very short topic. And I have put possible future questions in the future that IB might put. The first one you can see that I explained because I really want you to know. But the other thing is they can talk about is the outline the full process needed for spontaneous origin cells. That's on the lecture. Go there. The answers will be there. What are the two properties of RNA that it's so important? Outline why fatty acids are likely to be the first components of the early cell. All of these questions, believe it or not, they are answered in my lecture. So if you go to my lecture, you'll be able to answer them. And thank you. That's pretty much for this.